get to it. So a few weeks ago, we talked about photosynthesis. And we talked about the reaction. It's one of the most important biological reactions. It happens usually by primary producers, such as plants, algae, and some bacteria. These guys can be found on land, so trees undergo photosynthesis, algae in the ocean undergo photosynthesis as well. And within these primary producers, you have these specialized cells called chloroplasts, and that has the pigment chlorophyll. So these are actually the two cells in the pigment that actually create this photosynthetic reaction. And again, like we learned, we have photosynthesis that takes carbon dioxide, which is CO2, and water, which is H2O, plus sunlight to create glucose, C6H12O6, and oxygen, O2. Glucose that we learned last week is a monosaccharide. It's a simple sugar. And today we're going to get a little bit more into how this actually gets broken down by other animals and other microorganisms through cellular respiration, this glucose molecule here. So like we also learned a few weeks ago, cellular respiration is sort of the opposite of photosynthesis. This is where, living animals, other multicellular things, even some single-celled bacteria, take the sugar from photosynthesis plus oxygen and turn it back into water and CO2. So we learned that cellular respiration is sort of the reverse and the opposite of photosynthesis. And this kind of like cyclical reaction chain is what sort of makes your ecospheres tick and what makes them go. So today we're going to focus on the second half of respiration and what really goes into that and how multicellular animals as well as other microorganisms break down the glucose and the sugars that photosynthesis creates. So the first step in the cellular respiration process is that glycolysis breaks down the glucose into pyruvic acid. Glucose is not that well used by cells. Pyruvate or pyruvic acid is a much more readily available molecule, so cells are allowed to use that a lot more easily than they would glucose. So glycolysis just breaks down glucose into pyruvate or pyruvic acid. After this occurs, there are two forms of respiration that occur within the cells. There's either aerobic or anaerobic respiration. In aerobic respiration, this happens in the presence of oxygen. And this creates around 32 ATP molecules, but overall there's a net of 36 ATP molecules in aerobic respiration. And I'll get into what that is in just a second here. The other form of cellular respiration is without oxygen. So this is anaerobic respiration, also known as fermentation. And this mainly happens in microorganisms, but a little bit some of our cells, some of the multicellular animals do that as well. And this represents an incomplete breakdown of glucose and only provides 2 ATP molecules per molecule of glucose. So a lot less ATP is produced when you undergo anaerobic respiration compared to aerobic respiration. And I keep using this term ATP. Well, ATP is really the energy currency of the cell. This is where cells get most of their energy from. ATP is shortened and actually stands for adenosine triphosphate, but we'll just consider it ATP for this class. So in respiration, again, we have our glucose and our oxygen that breaks down into H2O, water, CO2, plus the 36 ATP molecules that organisms actually use for energy. So here's another little schematic of what happens after glycolysis, where you have oxygen absent. That's fermentation or anaerobic respiration. When you have oxygen present, you have aerobic respiration. And the process of glycolysis, breaking down glucose into pyruvic acid, also creates ATP, some energy as well. So this is why there's 36 ATP molecules as a net of respiration. So here we go, a little bit breaking this further down. Again, we're aerobic respiration, oxygen is present. Pyruvate is further broken down in the Krebs cycle. Energy released from the Krebs cycle is transferred to the electron transport system. And then this electron transport system uses oxygen as a final electron acceptor, producing 32 ATP molecules for every 1 glucose molecule present. And this entire process takes place in the mitochondria, which is the powerhouse of the cell. So this is a schematic here to help sort of break that down rather than just have words on a PowerPoint. So we're starting here. Again, this is aerobic respiration in the presence of oxygen. You have glucose gets broken down to pyruvate via glycolysis. This itself creates 2 ATP, 2 energy molecules for the cell. Pyruvate then gets transferred to the Krebs cycle, gets further broken down, and this further breakdown of pyruvate in the Krebs cycle creates 2 more ATP cells. However, aerobic respiration does not end there. Electrons and oxygen created from the Krebs cycle then gets transferred to the electron transport chain, and then this final breakdown of energy creates 32 ATP molecules for the cells to use. So in total, aerobic respiration creates 36 ATP. 2 plus 2 plus 32 is 36 ATP molecules. Any questions on that so far? I know I'm throwing out a lot of words at you pretty quickly, but this is sort of really the only way I can explain it. But please, if you need me to go back, please just shout it out. I'm happy to do so. So we're all good on the process of aerobic respiration, how it works, the different steps, how much energy it creates, and who does it. All right, cool. Now the flip side of that is anaerobic respiration, again, where no oxygen is present. Glucose is broken down, again, in glycolysis into 2 pyruvate molecules and 2 ATP molecules. However, because there's no oxygen present, there are some harmful byproducts when pyruvate is broken down. In animal cells, in multicellular organisms, this is lactic acid. This harmful byproduct can be expressed as lactic acid, which is sore muscles. If you ever worked out at the gym, experienced sore muscles the day or 2 days after, this is just a lactic acid buildup in those cells. However, in some microorganisms, pyruvate is converted into alcohol through fermentation. So this is why we're doing this today. It goes into your sourdough starter project coming up next week. So in microorganisms, if there's no oxygen present in anaerobic respiration, this is also known as fermentation, and a byproduct of that is alcohol. And again, anaerobic respiration is way less efficient than aerobic respiration and only produces 2 ATP molecules per glucose molecule. So way less energy for the cells to use if there's no oxygen present. And again, this is sort of a schematic of what this shows, the anaerobic respiration. You have glucose as the product of photosynthesis. Glucose is then broken down through glycolysis to create pyruvate. In yeast cells and other microorganisms, this anaerobic respiration is known as fermentation, where it creates alcohol as a byproduct. In animals and bacteria, this creates lactic acid as a harmful byproduct. Because there's only 2 ATP created in anaerobic respiration, this is not efficient for us to use. Without aerobic respiration, our metabolism would completely shut down. So we cannot live just through anaerobic respiration. We need that oxygen to help us sustain ourselves. Again, final review here, glucose into 2 pyruvate through glycolysis in itself creates 2 ATP molecules in aerobic respiration. In anaerobic respiration, it creates 2 ATP molecules, but that's it. That's all it does. And then in aerobic with oxygen, it gets further broken down through the Krebs and the electron transport chain within the mitochondria of the cell and notes 36 ATP molecules as a net use for cellular energy. So that's really all I have for you guys today as far as lecture goes. But I know threw a lot of stuff at you here relatively quickly. But if there's any confusion, please just let me know. So for next week, what's due is the sourdough starter project. Some of you have already sent me the start to your sourdough project. That's fine. No worries. You guys are a week ahead of schedule. If you haven't, don't worry about it. It's officially due next week. So you've got yourself the week to set it up. Hopefully you've got all your materials already. You don't need to scramble to get those together. What you want to do is you want to sign up at this website, scistarter.org, Sourdough for Science. Create your own little profile. This website has a ton of great videos and information for you guys to use on what you'll be doing. It's also found in the lab manual, kind of a step-by-step guide on page 22 that it starts. This whole experiment will take roughly 2 weeks. So it's a 14-day experiment, but it requires attention every day. So every day you're going to measure your flour and your bacteria and your yeast and figure out how much it rises. I'm going to wait for these guys here to stop lawn mowing, blowing leaves everywhere. So make sure you're at least taking data every day on it. And the website, got a question here. Yeah, you'll need to get everything that it says, the materials list on page 22. So all the materials are on there. Yeah, you need flour. I don't believe you'll need yeast specifically, but you do need flour. Any type of flour will work. Yeah. Yeah, so you'll use this website as a place to store all your data, and then you'll write up a little progress report at the end of the 14-day experiment. So make sure you're taking data every day, you're not falling behind. So it should be pretty simple. You're just measuring the height of it, but sometimes if it's so simple, it's easy to forget about. So this is the sourdough project that will be, you'll send me a photo of it to turn it into me next week. And then a few weeks from then, you'll do a little progress report on it. Any questions about the sourdough project coming up?
So like about the project, if you didn't start it already, like so, and then you say we gotta turn in the picture next week, right?
Yep, just send me a photo of it next week. I don't need a screenshot of your website portal. I just need a photo of the wheat and flour in the jar, so that way I know you've set it up.
Oh, so we just show a picture of like that we set it up then.
Yep, just show me a picture that you set it up.
Okay, thank you.
Yep. Another chat question here. Gluten-free flour is okay. Yeah, should be fine. I think you asked about that last week. I never got back to you. So yeah, gluten-free flour is fine. If it doesn't work, then maybe that's something you look into for your, maybe that's something you write up on and say, this is sort of something that I manipulated, and it would be cool to compare gluten-free versus regular flour if you have that available to you. Be a little interesting twist to this project, but yeah, you can use gluten-free flour. All right, yeah, I don't think there's any data sheets for this. Again, a lot of informational videos on the website, and your book in your lab manual does a pretty thorough step-by-step guide on how to set everything up. So that's all I have on the sourdough. So due next week, again, sourdough, send me a picture of everything set up for points. Not due next week, but you should be thinking or about working on is weekly measurements of your ecosphere. Hopefully everyone's ecosphere is alive and thriving. Take your daily data, look at how many plants are died, what's the water quality like, what's the pH, what's the temperature, all that stuff. And then again, daily measurements of your sourdough once you've started that. I don't need the daily measurements. I just want a picture of what you guys set up for points. Again, start thinking about ways to manipulate your ecosphere, keep a close eye on it. We'll be doing a little manipulation of that in a few weeks as well. So don't let that get behind. Keep monitoring your cool little ecosystem that you've created in your jar. I'm just going to make a quick note on late assignments here. So, first few weeks of the semester have been pretty relaxed about late assignments. This is my first time doing online class using Canvas. Everyone's getting their bearings sort of. We just had the add drop deadline passed last week. So new students were added to the class. But from here on out, all the late assignments will incur a penalty. So next week you have your sourdough picture due at 11 a.m. before class. If it's due after class, let's say 11 to midnight on the same day, then you'll receive minus 10 points. So the maximum you can get is 18 out of 20 for that assignment. And so you'll be deducted 10% for every day that's late up to 2 days, after which I will no longer accept the assignment. So make sure you guys are getting those in on time. So for example, turn in next week, you turn in before 11, you're good to go. The same day, but after 11, the maximum you can get is 18 out of 20. If you turn in on Friday, the maximum you can get is 16 out of 20. If you turn in on Saturday, the maximum you get is 14 out of 20. After that, I will no longer accept assignments. So make sure you're staying on top of it. Haven't had too many people turn in late assignments, but it has been an issue for some people in the class. So make sure you're turning in those assignments on time. And lastly, we've got a chat. Yeah, we can talk after class about confirming whether you've gotten stuff in to me or not. I know I don't email you guys back directly once I do receive your observations, but I try to have the grades updated on Canvas as soon as possible. So if you think that you sent to me your observations and your Canvas grade doesn't reflect that, please just shoot me an email. We can figure it out. Sometimes emails get lost. But yeah, we can definitely always, if you definitely want a notification to say that you've received it, please just shoot me an email if you're concerned about that. So exam 1 next week. It will be after our lecture. I've scheduled it for 12 noon on campus. It will last 30 minutes and it'll be around 30 questions long. I think right now I've got it at 28. It's going to be all multiple choice. So, but within that multiple choice, you have some true, false, and fill in the blank questions, but it's going to be all multiple choice. The exam is open note and open book, but not open internet. So have your notes in front of you, have your lab manual in front of you, but you can't be flipping to the YouTube channel. You can't be Googling. You can't have the PowerPoints open. Canvas can track all that stuff and track like what you looked away and stuff like that and where you're clicking. So open note, open book, but not open internet. Everything we've covered thus far, so including today, all week 1 stuff, week 2 and week 3. Yeah, I will send in the link to the YouTube thing after this. Yeah, so the question was, can you send us a link to the YouTube channel? Yes, I will. I'll put it in the chat here so everyone can see it. Another question was, will the exam be open all day or do we have a specific time slot when we need to take it? The exam will be open from 12 to 12:30, and that's when you need to take it. So that's within our class period. So make sure you're taking it at that time. It's going to be on Canvas. Hopefully there's no technical difficulties. I've never created an exam on Canvas, but if there is, we'll adjust accordingly. For any of you that have sent me stuff through the Student Success Center, you will get extra time. I'll create that for you. So if you've sent me that stuff, you're good to go. If you have not, please send that to me as soon as possible so that way I can add you into Canvas, saying that you get time and a half or extra time, whatever it is. So if you've sent that to me already, you're good to go. If you haven't, please do so as soon as possible. So like I was saying, everything that we've covered up to this far, including this lecture, is fair game. Mostly on the conceptual stuff. I won't quiz you on stuff like, how deep is the substrate layer in your ecosphere supposed to be out. That's the stuff I won't quiz you on. It's going to be all conceptual background information from the lab manual as well as my PowerPoints. Any other questions about the exam or today's lecture late assignments, sourdough starter projects, anything like that? All right, then I will stop sharing screen. You guys are good to go. I'll stick on if you want to confirm with me that your stuff was turned in. But other than that, you are good to go.
I have a question, Max.
What's up?
So I sent you because I remade my ecosphere, so I sent you the aquatic one. Is that okay?
I got that. Yep, you're all set to go. You got points for it. You're good.
Okay, thank you.
Yep.
Good morning, Max. I just wanted to make sure that you received the observations that I sent. Because
Yeah, let me check.
I know
Jesse, right?
Yeah, reaching
Cool.
out to
Let
you.
me see, Jesse. Yep, you're good to go, Jesse. Got them from you.
All right, I'll go ahead and save it because I know there was like two emails that was confusing me. That's why, but I'll go ahead and save that one in my
Cool.
notes.
That's the one that
thank you, Matt.
starts with the sea lavenders, right?
Give me a second. Sorry, it's been a long morning. Yes, it is.
Cool. All right, yeah, you're good to go.
All right, thank you, Max.
Would you mind, sharing the YouTube link?
Yes, I'll do that right now.
And then I also had another question about the algae project, which I know is in the future, but I was looking online to see which one I needed to order, and I was confused which one I needed to.
So if you go, I can also share the screen.
Yeah.
But here is the link to my YouTube channel. Make sure this works. You can go ahead and just click on that. And then I'll show you what the algae.
It works.
Did that work for you?
Yeah, the YouTube.
The YouTube link, you saw the videos?
Yeah, I did. Thank you.
Cool. And this is, I'm going to share my screen now. This is where this is algaeresearchsupply.com. Once you're at their homepage here, all you want to do is type in the search bar, SDSU, and you see custom products, materials for San Diego University. Okay. Click on that, add it to cart, and then once you're getting ready to check out, you can put your code in and it'll give you free shipping and free everything.
Okay, great. Thank you so much.
Sure.
Have a good day.
Yeah, you too.
All right, see you next week, Max.
Have a good one. Have a good weekend.